So Miles, if you wouldn't mind throwing up polling question number 1, we're going to ask our audience to participate now and tell us which of these best describes how you typically manage your company's regulatory needs and requirements. Do you handle all regulatory management and agency interactions in-house? Do you outsource regulatory to a consultant? Do you outsource regulatory to your suppliers? Do you take a hybrid approach, managing some in-house and outsourcing others? Or are you in the process of exploring the best way to manage regulatory? So take a minute to answer that question. We're going to post those polling results in real time here in a minute and have a brief discussion about it. But in the meantime, let's take one more. I'm moving around here. I'm pretty impressed with the questions that we're taking here. So I want to make sure that we address our audience's needs before I put my own in front of theirs. So let's take another audience question while the polling is going on. Let's take that. Jenny asked the question: comparability assessments with respect to analytical assessments. What pitfalls or lessons learned have you had when dealing with regulatory agencies? Were there any surprising gaps that needed to be addressed that were not thought of in planning or drafting comparability protocol? I want to hit that one, Dr. Chamow, because you mentioned comparability assessments a little bit earlier. I thought that might be a nice one to address.
Yeah, so let me just understand, Matt. The question is, have we experienced shortcomings? I'm paraphrasing here. In comparability assessments that have been thrown back at us by regulatory agencies?
I think that's a pretty, I'm looking at the same words you're looking at, so I think that's a pretty fair assessment of the question.
Okay. The answer to that is yes. So this is what I'm thinking of is a situation where we've done a comparability assessment on a program that this was a monoclonal antibody that we had initially produced in a certain cell line and then changed cell lines during clinical development, which, by the way, is the largest process change you can make in a biologic is changing the cell line. It's absolutely doable, but it is something that has to be done with thoughtfully, and again with this notion of comparability in mind.
Yeah.
And in this case, the issue that we ran into was, of course, glycosylation, which is going to be one of the biggest variables in changing a cell line, and we got variable responses from regulatory agencies of different countries about the importance of the changes that we were seeing. In some jurisdictions, the changes were minor, even though they were within our expected range. And then in another jurisdiction that I'm thinking about, the reviewer raised concerns about whether or not these are truly comparable. I mean, this is a judgment call. Reviewers at regulatory agencies they may be reading the same application and the same text, but based on their own experience, they're going to have different interpretations of how important this is.
Yeah. Dr. Pluschkell, you've mentioned to me that while we're talking about comparability assessments, you've kind of lumped comparability assessments into things such as stability studies and other activities that can't be easily or efficiently revisited or redone. So give us some thoughts on that while we're talking about it. What's your advice on putting those activities on sound footing from the outset? I know it's an unfair question because it's a big challenge.
Well, that's what we're here for, right? So I think number one, first of all, I think there's an interesting anecdote I can share, right? Number one, though, a comparability protocol, and I don't know whether Jenny was thinking of it from a clinical or commercial product perspective. In the clinical setting, there's not an absolute requirement for you to send in a comparability protocol to the agency. You could principally choose for certain changes to just make the changes and then send in the amendment, okay? So it's a choice for you to make. I would think the choice of sharing with an agency your plan for how you're going to assess comparability depends on how complex it is. The more complex, the more likely you might benefit from running it by the agency first. Okay, you may have one internally at any rate. But here's one example that connects stability and comparability that I've seen as a real pitfall. If you send in a comparability protocol and you are saying that you're putting the post-change product on stability to confirm its comparability, the agency will hold you to it. And you just put a certain time frame for waiting in there. You can hardly take this sort of thing back. So just keep in mind that when you are writing a comparability protocol, that you're writing it in a way that you feel is consistent with what is needed. If you don't feel that that the stability data are needed to show comparability, then don't say it. Okay, because if once you say it, the cat's out of the bag, and I have seen products being put on clinical hold because you're doing a comparability assessment and you said you're going to generate 6 months worth of stability data, and if you don't have them, they may say, well, you told us that that is needed to show comparability, so where are the data, right? So you got to think it through and think about what you think is needed to get into the clinic again, right? Presuming you were in the clinic before and you continuing to be in the clinic, versus what is data that you gather alongside, right? Concurrent with a clinical trial, such as stability data are always required, right? Concurrently with the clinical trial. And that's also one plug that I would make, because you will inevitably have some changes during CMC development. Don't forget the retains. When Steve says you may not need a cell-based assay now, if you can have retains to later on show the alignment in potency when you're making method changes, it's worth gold. If you don't have the retains, there is no way to go back. And comparability has become, over the years, in my perception, really more stringent than it used to be. So when, in a way, I would say it used to be that suitable for use in early clinical trials was good enough. Now, more and more often, there is almost an expectation that what you put into the safety studies, preclinical safety, is nearly, I don't want to say identical, right? But truly comparable to what you put in the clinic, truly comparable to what you put in subsequent trials. And that's different from just a perspective of suitability for use.
Yeah.
I think that's really true. I want to emphasize that point. Yeah, Steph, you're absolutely right. And I think what has happened is, we in the industry have demonstrated that we are capable of doing this, that it is possible to make comparable material, and that we have the analytical methods in order to demonstrate that comparability is multifaceted. And I think what regulatory agencies are doing is they're just saying, well, you guys can do this. So the expectations have changed. They're not what they were 20 years ago.